in microscopy. What are the major causes of error in urine chemistry? There could be transcription error, there could be sampling error, there could be contamination error. But in this case, if you have a well proper labeled sample, you have to just keep those samples in the instrument. Instrument will do the work on the next stage. Then there could be a question that, is there any rate of chance that we have to recheck and review the sample? AI has only one quality, that whatever will you guide the instrument will follow the same. But it has some limitations also. If you will run the 100 sample, after 100 sample, I think most probably you have to view 1 or 2 sample, that only for a single or 2 times, to check either the instrument is giving you the same review or not. But if you have any image or which you have seen in the microscope, if you have any image which will be available in the microscope which is not available in the system, you can check those images, collect those images and put in your pen drive. Same pen drive can be injected within the software, and software will capture it in the further way. For neonate samples, if sample volume is less, you have to centrifuge the sample, you have to make the slide, you have to do the biochemistry test, you have to do the microscopical test of the sample. Sometimes the sample volume may be a differing parameter. At that time, this instrument will already require 900 microliter of the sample. You didn't need a large volume of sample for ACR or any other parameter. All the parameter can be done in 900 microliter of the sample. Data management. You can access the data till 10 years or 50,000 data can be stored within the instrument, and all the data can be taken with the backup module. There are 3 modes in the instrument. If you have to do the biochemistry test of the urine, if you have to do the microscopy of the urine, if you have to do all the biochemical tests and microscopy of the urine samples. The modes have been selected on the basis of the requirement of the lab. If you have to do only the biochemical test, the CPT will be different. If you have to do the microscopy and biochemical test, the CPT will be different. That's why the modes have been selected. You will be given racks with the instrument. There is no limitation that you have to use same tubes and different kinds of tube or containers for collection. Any tube can be available which will be sterile, can be used for running the samples. The throughput of the instrument is 125 samples for only sedimentation mode. For chemical study it is 160 samples per hour, and for hybrid mode it is 125 samples per hour. These are the parameters which you can see within the instrument. Other than the biochemical analysis, specific gravity, leukocytes, nitrates, pH, protein, glucose, ketones, urobilinogen, bilirubin and blood, you can see other parameters also. In RBC, you can see dimorphic RBC, then normal RBCs, hyaline, cast, crystals. Each and every parameter will be available within the instrument. This is how the instrument works. The sample has been taken. There is no centrifugation, there is no mixing of the sample outside the instrument. You have to just keep the sample will be homogenized within the instrument. The probe will touch the sample, homogenize it and transfer it to the cuvette. After filling it into the cuvette, gravitation force immediately act upon the sample. As the gravitation force is applied, the higher the size, higher the weightage, the sample will flow at the bottom. Immediately, simultaneously, the digital microscope will read the sample, and it is best for all the diagnostic institutes and the technician who can continuously and simultaneously observe the sample also on the screen. If you will see this one on that screen, you can continuously monitor the sample, either it is giving right or wrong. This kind of screen you will be able to see in the instrument. Thank you.
So nice presentation, Mr. Sunny. I think this is an important in urine analysis. What I got from this is, what is important is data storage of this sample, because we are not storing the data for the urine samples. This is important in this machine, and 3 modes that we are seeing, or the microscopy and all, which is important for our postgraduates. They need to see the crystals, the casts that the clinicians want us to see in some renal diseases and all. So important in this instrument.
Any question?
Any question from the audience?
Anyone can ask the question, please. Gravity segmentation. Sir, we have used a gravity platform below the cuvettes. When the sample is filled inside the cuvette, gravitational forces are applied. Because of the gravitational forces, gravitation can be applied on any living, non-living thing, anywhere. Gravitational forces are applied, the sample will fall on the gravity because of their sizes. As the sizes are different, they will fall at the bottom. At the same time, the contrast microscopy will read the sample. And instrument already have images for these cells. You can use any kind of RBC which you can be able to see in the microscope. Same images are already within the instrument. As the cells passes through the digital microscope, it immediately defines the sample is pus cell or RBC, cast, crystal, whatever the cell will be, they will be immediately captured, and same data has been converted from calculatory to digital on the screen.